Right, I am. I'll wait a little bit to see if anybody else gets on here. I don't understand. Here we go. Is it not gonna work or okay. Let me just wait 1, 2 more minutes. Let's see. Pay no attention to the man behind the curtain, okay? Just saying. Trying to get stuff out of you guys' view. No, how I got to that screen. Not that. All right, so we're gonna go ahead. Um, it's not like lighting up that it's making like recording my voice or anything. I hope that you guys can hear me okay.
I can hear you.
Cool. Yeah, that little like microphone symbol at the top, like usually it'll go green or whatever when I talk, but it's not doing it, but whatever, as long as you can hear me. Um, let's go into week 5, where you're probably going to hear my little chiming go off. I can't figure out how to mute it without bringing it up in front of everybody, so it's just going to keep going, but it's okay. Everybody's very excited. It's basically our Friday, so they all want to chat. So week 5 deals with chapters 6 and 8. So we'll go over chapter 6, the genetics chapter, and we're going to go over chapter 8, which is going to be the genetic engineering chapter, which I realize that if you're on here and you're in my night class, that we haven't gone over it yet, but hey, consider it a primer then, I guess. So we're going to go ahead and let's just go review those. We'll start with chapter 6. This is the information that we were expected to learn for chapter 6. This is, when I put these up here, it's the stuff straight from the study sheet. Sometimes I'll like kick out some stuff that I don't really want to worry about at the time, but chapter 6 is pretty heavy. So there's just a lot of stuff on there to know. Um, like knowing about what a genome is, what the genotype versus a phenotype is, the structure of DNA and its nucleotides, the anti-parallel strands versus when we are replicating DNA, the semi-conservative aspect, which means that we'll start off with one double strand like that, whatever, here's your ladder. And then when you're going in to make the two, to turn one into two, right? Because we're replicating, we're duplicating basically. One side is old, the other side is old. And then, because I don't have my thing ready, here we go, here we go. And then whenever we do go through, I can't see my cursor at all. It's really killing me. Here it goes. New, and then new, and using the old ones as a template. And that's what makes it semi-conservative. And that's what this is talking about here in the study sheet. When we talk about replication, we're only talking about DNA replicating itself into DNA, such as whenever you're growing a new cell. And that's really the time that you would be doing it, is by growing new cells, whether you're growing a whole person from an embryo, or if you fall and scratch your knee, you need to repair your open wound, you're going to need more cells for that. So anytime that we're doing that, you're going to need DNA replicated. Okay, whenever we're making our DNA, we have the replication fork, essentially, as we're opening up our DNA. Jeez, I know that I have a cursor. Here it is. If this were your DNA and you're trying to open up and move in this direction, this would be your 5', just saying, towards the 3'. And this, we're adding on these nucleotides as we are going, right? So that's the leading strand. And then the other strand is getting opened up gradually. So we can make it in little fragments as it's opening up. So as this opens up even farther, we'll make another fragment. These fragments, again, and again, that's the lagging strand. That checks, right? Like we can all kind of get on board with the lead one being the faster one, basically, because we have to be able to add our nucleotides in that 5' to 3' direction, always 5' to 3', always 5' to 3'. So then this would be 5 over here and then 3 over here, always. So now we have our setup of what our leading and our lagging strand are doing, and that lagging strand, while we're making that, will be little fragments. And those fragments that we have made here, like we said before, each time you're building off of, making a new fragment further up and further up and further up as you're going deeper into that fork, you have to add it on to something. And that was the same whenever we started on this one, initially. You had to add on to something. Those are our primers, and they're RNA-based primers when we're talking about replication of DNA. We'll go back and replace them with DNA later on. But for the time being, you're getting these fragments. And because we have these fragments, this is not useful for DNA. It has to be in a big old long straight line, because when we open it up to go look at our genes, we can't have just pieces. When we open it up to recopy it later on, we can't have just pieces. It has to be one whole big old piece. There's an enzyme that actually seals together those fragments, and those fragments are called Okazaki fragments. And the enzyme that seals is called DNA ligase, which if you have had a chapter 8, then you understand that it's pretty important for definitely for genetic engineering, but obviously for DNA replication as well. So that's a pretty good little primer over a pretty big concept in chapter 6. When we move on to RNA, DNA to RNA is called transcription. So if we were to look at this, Weird, I pressed E and I didn't mean to press E. DNA to DNA, again, just to remind everybody, that's called replication. But if we're going DNA to RNA of any kind, this isn't just messenger RNA, okay? This is any RNA. So whether we're talking about, you might be, what else is there? Remember, there's going to be messenger RNA. That's the working copy of your gene. We'll just put working gene, you get it. It's a working copy of the gene. rRNA, which is the structure of the ribosome. And then tRNA. And it's called transfer. So it's transfer RNA because it's going to be transferring the correct amino acid when we go through translation later on. So our working gene is mRNA, whatever. So this process going, anytime you're making RNA, had to come from a DNA template. That process is called transcription. Or it's like script or writing, same language. And then going to here from RNA, such as the message encoded by our messenger RNA, that this would be our translation, translating the nucleic acid language into the amino acid language. And that's going to make proteins. So that's the idea of how that's going to work. And when we're going from DNA to RNA, that process is going to occur in 3 steps. There's the initiation. And this is useful because these are the same 3 steps for transcription and for translation, but there's greatly different things going on in each of them, right? So first step is always going to be initiation. Second step is going to be elongation. Third step is going to be termination. Now, when this is happening, when we're transcribing things, so going through transcription, let me get a different color here so we can actually see like what I'm going to be writing here in a different color. Go back to red. All right. If we're going through transcription, this is essentially going to be relying on the RNA polymerase. So DNA gets its own polymerase whenever it is replicating, right? RNA polymerase binds at the promoter. So that's like our initiation. We're going to open up the DNA, double strand, bind our RNA polymerase at the promoter, and that's initiation. Then we can add on our RNA nucleotides that are complementary to the template strand of DNA, so that the message matches the coding strand. But we're using the template strand as the template to make the complementary nucleotides match up to carry the correct signal on the messenger RNA. Well, that's great. So we did that, and we can get to a point eventually at the end of it where we're at the end of the gene and the sequence isn't matching up the way that it would be expected to. And there's other like molecules involved in this that you don't have to learn about, but it will terminate. Just gets to the end and it terminates. So that's pretty easy as far as how that's going to be working. So now we've got our messenger RNA, which is our working copy of our gene, and we're going to make it into a protein. So in translation, now we're going to have the ribosome. This is our initiation, okay? Number 1, for initiation for translation. We'll clamp on or bind onto the messenger RNA. The next step for our elongation, we will be bringing in tRNA that is attached to an appropriate amino acid. And what do I mean by appropriate? that tRNA has an anti-codon that matches the codon that is on the mRNA. The codon is on the mRNA. Okay, so the mRNA is nucleic acid in RNA form, ribonucleic acid. And it's just nucleotide, 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 nucleotide. And every set of 3 of those nucleotides is a codon. That's the word for those 3. Okay, so if you were to look at those same 3 nucleotides, they're, match up in the DNA. They're called triplets in DNA. But that's not even a functional thing. It just would, if you were looking at it, helps you figure out what you're talking about. But in translation, that messenger RNA, the important thing about it is the codons. So whatever those 3 nucleotides say, AAA, brings in the matching tRNA based on the anti-codon sequence. So what I mean is hey man, I can't see my thing. Okay. If we are talking about, your codon reading AAA, your anti-codon would then be U, and it's attached to that ridiculous T shape. This is like a squatty version of it. And then at the top of that T shape, you would have an amino acid that corresponds to whatever AAA codes for. All right. And you're not expected to know those. That's not something you are expected to know for the class. But this is just, you can look it up and see it so you know what's going on. You're aware of the possibilities, but you don't have to remember what AAA is for or anything like that. Okay. Cool. So that's basically how that would be working as far as transcription and translation. When we get to termination on this one, termination means that you are getting to a stop codon. And a stop codon is a 3 nucleotide. Codons are always 3 nucleotide, right? On your mRNA. So this is a 3 nucleotide sequence on your mRNA that does not code for an amino acid of any kind. There's no tRNA with an anticodon that matches it. Because of that, that's how the ribosome knows how to disengage, and you're done. So we call those stop codons, like UGA or whatever that might be, the codon or whatever, we call those stop codons nonsense codons also, because they don't code for an amino acid. There's 3 of them, those 3 stop codons, and everything else codes for an amino acid of some kind, okay? So that's the gist of transcription and translation. I wanted to cover it just one more time without having to introduce these topics as though they were brand new so that you could understand maybe a little bit better hearing it the second time through. I know we're already a little ways in, but I didn't want to miss out on that. Okay, so I'm just trying to make sure I'm not missing the operons, y'all. So the operons, I know you hate it. Don't we all? So the operons, I forget that it gets rid of the thing that I was drawing. So we have inducible and repressible. If it can be induced, it had to start out. It can be induced versus it can be repressed. If it can be induced, it had to start out as off, right? If it is off, that means, what does that mean? That means that RNA polymerase, which usually would be making our working copy of that gene, that it cannot bind. Or it can't, at least it cannot bind, or it can't do its job. It can't work. It can't elongate. Put that. Since we need to know those 3 terms anyways, those are terms you have to actually know, not just the concept. So like the initiation, elongation, termination. So RNA polymerase can't elongate, can't do its job, can't make the messenger RNA. So that's what's going on. We'll say what changes here. So because they're going to be different. What changes is that we're going to be triggered, or what are triggered, there's a term for it, induced, right? By something called an inducer. So the inducer will bind with, okay, whatever you guys know, binds with the protein that is blocking. So we'll even put this on over here, because a repressor, so this is the name of the protein that's blocking it. So this case, when we're inducing, that's just sitting there. That's how come it's off. All right. So it's off because of that repressor sitting there. So what happens is we're inducing it with our inducer, like we talked about with the lac operon. So lactose is our inducer. So it's going to bind with that repressor. And that's going to change its shape, and it's going to go away. And now we can start making those genes. So we'll just say, so it leaves. When it leaves, now RNA polymerase can do its thing, right? Because we don't have a repressor there anymore. All right, so the other guy here, the repressible operons. To watch this, because there's another screen. You guys are looking at a different screen than I'm looking at, and when I'm drawing it, it's making me, I don't know, double vision or something. It's weird. Okay. So how is the repressible going to be starting? Well, it's going to always start as on. How can you turn it off if it was off, right? Your only options are on or off. So what is causing this whole interaction? Well, we don't have any repressor. We don't have any, anything blocking our RNA polymerase. So the reason that it's on is there is RNA polymerase isn't blocked. Okay. What's changing about this? And the change, a repressor, it's also a repressor protein. It's always the repressor protein that's changing the situation. Okay. Either something is causing it to come off, or something is causing it to go on to it. So in this case, we're going to take our repressor that's just out chilling, and we're going to call it into work. And that repressor is called into work whenever usually we have an excess of product. Something you're making, your cell isn't using it up anymore. And if it isn't using it up, it's going to build up. And when it builds up, it's going to bind with that repressor protein. Okay. And now, just like before, so this is activating that repressor, this inducer, and say it binds with the repressor so that it leaves. So we can say deactivating on that top one. So that you can relate these two, okay? So our repressor on the bottom, we are activating it by our excess product, and that will block RNA polymerase. So we're not making any more of the mRNA, and therefore not making any more of the proteins involved in whatever process, right? No matter what, okay, no matter what is this? Is going on there in those operons. Remember, these are called operons. These are only in bacteria. Okay. So that you know it's not related to that, to this part only. But right. So these repressors always bind at the same spot on the DNA. It's always right next to that RNA polymerase binding site. Remember what that's called, the promoter. Right. So RNA polymerase binds and starts its work at a promoter. I know we've said it before, but I just want to reiterate it. And then we have the repressor. When it is binding and blocking, whatever situation had it there, whether you are inducing it or whether you are repressing your operon, the repressor always binds at a site right next to the promoter called the operator. And that's pretty much it for operons. So I hope that helps a little bit with the operons. We also talked about the different kinds of mutations. I'm not going to go into that too much just because I feel like the tables and explanations that are in the text as well as in the slides are probably just fine. But remember, missense mutation, you have nucleotide change or changes that lead to a different amino acid. Nonsense mutation, you have nucleotide change or changes that lead to no amino acid being made, aka stop when it shouldn't be there. And then the other one was a silent mutation. And the silent mutation is whenever you have a change in nucleotide that still codes for the exact same amino acid. So you don't see any change in the protein's primary structure, which is its amino acid sequence. You don't see any changes primary structure, and likely then, of course, wouldn't see any change in the secondary or tertiary structure. So that preserves the functionality of that protein, even though there was a nucleic acid change. So that's important that we can recognize there was a nucleic acid change, but still maintain our amino acid integrity, if you will. Now, the other thing was the back mutation, which is just going back to what the original was. This is more applicable to like bacteria and stuff that divide faster than we do. Okay. Cool. I just want to be sure that I'm not missing anything in the chat or something. So if you guys have any questions, let me know. You can always just yell it out, or you can do chat. It, for some reason, doesn't always pop up on the chat. So if somebody does see that it did and I'm just ignoring it, please let me know. Oh my God, I'm getting these chats though, that's for sure. Dr. Morford and Tim. Okay, so chapter 8 is the genetic engineering chapter. We talked about some major concepts in here and then some like kind of smaller ideas. We got into restriction endonucleases that cut at those palindromic sequences. Remember, like race car, forward and backwards, except here we're talking about DNA. Palindromic sequences, and they cut in such a way to create sticky ends that are overhanging. So single-stranded sticky end and another single-stranded sticky end the other way. They normally would match up to each other, but we're going to engineer some DNA that wants to stick in there instead. And so that's the importance of restriction endonucleases in genetic engineering. CRISPR is a useful, CRISPR-Cas9 is a system basically in bacteria that they use to get rid of foreign DNA. Same thing that they do with the restriction enzymes. But we can basically design it to cut at any DNA sequence ever. And we can make it specific as well. So that is a useful tool that hasn't been, adequately used so far in science, but I think it will get there eventually, but it's not now. It's not now. All right, then we got into PCR, which if you guys were in any of my classes, and you guys in the evening will probably see that I will go over that in probably the most detail, but this is because PCR really is the #1 most used diagnostic tool in hospitals, as far as not even just like infectious diseases, but definitely infectious diseases, but also with diagnosing certain cancers and stuff like that. We can look at genes and see, do you have this version of a gene or not, using our PCR as part of that process. PCR, all that PCR does, by the way, oh my God, I have, there it is, okay. PCR, all it does, it just duplicates DNA every round. So doubles the DNA for every cycle. All right, so that's all PCR does. So its purpose is to amplify DNA. That's what we would say. It's got 3 important steps. I know it has 4 bullets, but really, if you look at it, it's only 4. It's only 3. 3 steps. So the first step, it says heating will denature. So your first step, of course, to summarize this, number one is denature. Anytime you hear denature in biochemistry in any way, shape, or form, which, hey, I hate to break it to you, this is biochem, right? So anytime you hear denature in biochem, we're taking something from its intended usual working state and turning it into where it doesn't function. So we call the nature, the natural state of this is the functional state, and the denature is taking that natural state out of it, making it non-functional. So in this case, we're heating up DNA, separating those strands, which were held together by hydrogen bonds. And then we can have our primers combine. Remember no, there it is. This step is called the anneal step. That means to stick on the primers. Anneal is sticking something onto it, so prime. We'll put it there so we don't forget it's the primer step. We cool it down a little bit so that our primers stick on. The primers are in excess by majority, like really in excess compared to the DNA concentration in there. So that way you can ensure that your primers are going to bind on there and serve their purpose. The primers are designed as 15 to 30 base pairs long. So 15 to 30 would be like A, T, C, G would be 4 base pairs. A, 1 base pair, 2, T, 3, C, 4, G, you know what I'm saying, right? Anyways, these are 15 to 30 base pairs long, and they are made to bind to specific areas around a target gene so that you can identify something if you are adding on nucleotides. So if your primers are that specific and they're only going to bind whenever that sequence is there, and it will not allow elongation then if the primers don't bind. Therefore, if the primers bind, you can build off of those primers and make copies only if that gene is there. So if you don't have COVID and therefore don't have COVID DNA in there, then the primers aren't binding to anything. And therefore we are not building off of them at all. They're just chilling in that solution, no matter how many times you heat it up and cool it. You have to have them to match up in order to say. So if you were running that cycle and you didn't get any amplification out of it, that person would be negative for whatever you are looking for. Okay, so anyways, obviously then I feel like the third step is pretty, it's always farther than I think it is. Elongation, adding the nucleotides, it's the same thing, so elongate. All right, and then you just do it over and over again. So that would be one cycle there. And then doing it again is the second cycle. And then again would be the third cycle. And each time you are doubling what was in there in the immediately previous cycle. All right. Okay, so cool. So we use gel electrophoresis as a means to separate out different sizes of DNA fragments. The smallest ones travel the fastest. The largest one gets stuck behind, kind of like you can imagine if you were like in webbing at like a ball pit or something like that. And, you're trying to climb through all this webbing, but the balls can just like fall through, right? Because they're tiny. It's just like that. So that is the idea here with gel electrophoresis. And remember, DNA has a negative charge to it because of the phosphates in its backbone. So it's going to go towards the positive end. So we can use all of this information and all of our knowledge about bacteria and other organisms to utilize these things to our advantage through our cloning mechanisms. And we get to learn a little bit about single nucleotide polymorphisms, which includes having one nucleotide change in a gene, and that can cause potentially devastating effects or just like different effects, different traits. There's a lot of different versions of genes that can depend on just one nucleotide being different. GMO and transgenic is the same thing. That's just any organism that had its genes combined with another organism's genes. So their genome is now modified with another organism's DNA. Gene therapy is a concept where we are using viruses like HIV, we say viral latency, right? When they hide out in your DNA, like HSV, herpes simplex. And it will usually stick its DNA into your DNA. Well, we can use that mechanism, use the virus how it would normally do that, except have it stick in the genes that we want it to stick in. for example, a functional gene that codes for, working protein for somebody that couldn't make that. Normally they had a mutation that could have been deadly or something like that. This can be occurring in somatic cells, which is really the only way that we have it functioning currently. Somatic cells are your body cells. So as an adult, you could have these procedures done to help fix your faulty genetics. But if you want it passed on to your kids, then it would have to be a germline effect that affects your gametes or embryos. And that would be heritable from then on, from that baby or individual or fetus or whatever, from then on. So that's what we learned about in chapter 8. So let's go over our commonly missed topics. Start with chapter 6 then. Okay, so chapter 6, most commonly missed topics. We're going to start with semi-conservative DNA replication first. And what that is talking about is starting off with that original. This is just like we saw in that other slide. And then whenever you go down to separate it to make a copy, your old strands, original strands, have separated apart, and then you can add on your nucleotides, of course, in the 5' to 3' direction, just to remind you. Matching up to those. But you can see if blue represents the new guys that we're adding on that are complementary to the red old guys that are working as our template, then each new daughter strand, which we have 2, right? We have 2 daughter strands here. That's the whole point of replication. And those 2 daughter strands each consist of 1 old and 1 new strand. That is semi-conservative. So this slide basically goes through a little bit more detail on what that means and how it works. And as per usual, I'm not going to read through all of these slides for you. These will be here for your reference for you guys to look at, but this is just me trying to go through and put it into live terms. So a working live person is explaining it to you and not just reading it blank on the screen, because I know how that can be different. So we are conserving our original strand, but we're getting the other side of it to be new, and that's why it is semi-conservative. And then it goes on to explain on here, imagine the original DNA molecule as a zipper with 2 interlocking strands. During replication, the zipper separates into 2 single strands. Each original strand serves as a template to form a new strand. So we have 2 new zippers. Half of the zipper is originally there in the first place. The other half was the new ones. Okay, so that's all that's going on there. It's the same concept here. And our little mnemonic here is semi-C, which should be standing for same end, mixed in copies. So, and our little, I guess we could say, explanation of that. I have something in my way, hold on. Same, we're going through same end, mixed in copies. Same refers to the fact that each new DNA molecule retains one of the original strands, right? End will be how replication maintains the end or the structure of the original strands when it is replicating. Mixed is indicating that each new strand is going to be an old and a new together. The N is going to be the process of incorporating the new strand with the old strand. And the copies is just talking about a copy. You're making a copy. Okay. So I don't know if that helps, but there it is. That's our little memory thing there. So our questions associated with this concept: which of the following accurately describes the semi-conservative model of DNA replication? So our choices: both strands of the original DNA molecule. This is A. Both strands of the original DNA molecule are completely destroyed during replication. Well, if you're paying attention at all, then you know it's not that one. Each new DNA molecule contains 2 newly synthesized strands. Well, it's not, because it's semi-conservative. It's the whole point, is semi-conservative. So it's not 2 new on each one. Each new DNA molecule consists of 1 original. Okay, now we're talking. And 1 newly synthesized. Okay, probably this one. And then DNA replication produces a single strand of DNA from an RNA template. That's definitely not what we're doing. That's what reverse transcriptase does. So it's not that one. So C is our answer. The second question says, a researcher is studying a mutation in DNA replication that affects the accuracy of the copying process. The mutation results in increased rate of incorrect base pairing during DNA replication. This means, just so that you know, this would mean that your A's and your T's wouldn't necessarily be binding to only A's and T's. Same thing for the G's and C's. Okay, so there's errors being made. Which of the following aspects of DNA replication is most directly impacted by this mutation? So is it going to be having to do with the separation of the DNA strands? And it says by helicase. Don't have to worry about what it means. It doesn't have to do with the separation of the strands, right? That's not the problem here. B, the synthesis of a new strand complementary to the original strand. Now that might be it, because what this is saying is our new strand might not match up. So you're old, and as you're making your new alongside it, adding a nucleotide, and so on and so forth, even though this one might say AAA, this one might say, T, 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 but then there might be some sort of mess up where it's supposed to be G, and instead of having a C, it has another G here. Now, suddenly that doesn't match up. That's could cause a serious problem, especially when that one goes on to replicate later on. So that's what that's talking about. So that could be a problem. C says joining the Okazaki fragments during the replication of the lagging strand. Well, you would have a problem if you couldn't join them, but that's not what this is talking about, is it? This is talking about having errors. So D says the separation of new DNA molecules into daughter cells. This is like when we're pulling them apart. Remember in PMAT, when we're talking about mitosis, and in metaphase and anaphase, metaphase lining them up and anaphase pulling them apart. That's what this is referring to in D, and that's not what's being affected here. What's being affected is the correctness of the sequence. We're having errors in the sequence when we're adding on our nucleotides. So that's what B is describing, and that's what the answer is. All right, the next thing is the difference in transcription between bacterial and eukaryotic transcription. There's a number of things we could go over to remind you guys about it, but remember that bacteria don't have a nucleus. That's the biggest issue here. Okay, almost all of the difference is going to be from that. The more important one is, so I'm gonna put it here so that we don't forget. So guess where all of this is happening in the bacteria? This isn't happening in the nucleus. This is happening in the cytoplasm. The cytoplasm is just the goo. Don't get too confused by that word, cytoplasm. It's literally just the goo, okay? That everything's happening in, that water-based stuff, everything's going down in. So bacterial transcription is going to be happening in the cytoplasm, not in the nucleus, whereas eukaryotic is going to be happening in the nucleus. It has to, because that's where your DNA is kept. That's the whole point of the nucleus. So in bacteria, we have RNA polymerase. They only have one. It doesn't matter how many types they have, but that is a difference. They have different kinds of RNA, one type of RNA polymerase for all the different kinds of RNA that they have, whereas we have different kinds. Initiation, this is just all the difference. I made all the differences in here, but only really worry about the ones that are on your study sheet. But that would be the first one for sure, this one. Let's see, initiation, promoters, sigma. Oh, no, we care about that too much. Processing, and more. Okay, here we go. Bacteria do not undergo any significant processing. We'll separate these so you know where the marks are. Right? They're just making their mRNA and it's good to go. Whereas in eukaryotes, we do have to undergo processing. We have to remove our introns and join together our exons. Huge one, okay? So the nucleus versus no nucleus, that's a biggie. We'll put that one as number one. And number two biggie is going to be eukaryotes have introns and exons. And then you would, of course, need to remind yourself that those introns need to be cut out. We'll join the exons together to make a workable functional mRNA. Also, three, Only bacteria have operons. So that's the biggies, all right? I guess our little mnemonic here is bacteria copy simply, eukaryotes process thoroughly. So if you can remember that one, hopefully that'll remember you that, the details that are going on behind those statements. So our questions for this one, which of the following statements accurately differentiates bacterial transcription from eukaryotic transcription? Let's see what we got here. A, bacterial transcription requires multiple types of RNA polymerases, whereas eukaryotic, so this is hard because I just told you guys you don't really need to know this, and I put this in the question and I was just listing it off of information that I had that you guys don't need to know. So that will not ever be on your test, but that was in the original question. So pretend like it didn't happen. But yeah, it's not that. In case you didn't listen to what I said earlier about it, is that it's the other way around, if you were interested. So eukaryotic transcription occurs in the cytoplasm. We know eukaryotic transcription. That's DNA to RNA transcription. That, if you are working with DNA, has to happen in a nucleus, so it can't be this one. C, bacterial transcription involves very little mRNA processing, while eukaryotic transcription involves extensive mRNA processing. And this is true. This is basically the part where we're going to be talking about introns and exons. So that's the extensive that eukaryotes do, and bacteria don't need to do it at all. They don't have those. All right, and D, bacterial transcription uses, no, that's literally what I just said, that's opposites. So C is your answer. Okay. This is one that you would have only been able to eliminate A if I had put it on test. And like I said, I'm not going to do that. But if I put this on a test and hadn't proofread it for some reason, that you would have seen, hopefully that C would just be right. And you would have to eliminate A just because it's not right. So you would have to know better that way. But that's all you'd have to go off of. All right. So a new drug is being tested for its ability to inhibit transcription, specifically in bacterial cells. Which of the following mechanisms is the drug most likely targeting to achieve selective inhibition? So is it going to be DNA polymerase? Is it going to be the mechanism that shuttles mRNA out of the bacterial nucleus to the side? They don't have a nucleus. Well, we don't know for sure that it's going to be this. It's not going to be, but just FYI, we're going to go through the choices. Okay, RNA polymerase activity in bacteria by binding bacterial RNA polymerase enzyme. Or splicing of introns from eukaryotic mRNA transcripts. So it's not that one, because why would that affect the bacteria, right? So that's what this is talking about, something that affects the bacteria. So we know it's not B and we know it's not D. So now we're here sitting with DNA polymerase in bacteria or RNA polymerase activity in bacteria by binding specifically to the bacterial. So this is a lot more specific, I feel like. And if you see something ever like detailing it in a lot of detail like that, it's probably the right answer, because you have to go into detail to make sure that they know that it's the right answer. So if anything, you could delineate to that, but as it turns out, the DNA polymerases aren't different either. All right, so our next thing is the operons. And Lord, I hope that I have already gone over these enough for you guys. At this point, I hope that the previous explanation was good with our little chart making, but the repressible, they had to be off. So this is upside down from how we just talked about it, right? So if it's repressible, it's able to be repressed. So this one, sorry, I just said on and then I wrote the wrong thing. If it is repressible, it has to already be on first. And if it is repressible, because it is on, your RNA polymerase is working. That's what it means to be on. And so the change that's going to be happen in your repressible is activating a repressor. It's always a protein that's binding with a something. And then when it binds with the something, in this case, it will go and attach to its operator to stop the whole process. So that's the big, that's more of the words, but this is the big like summary of it. We can say repressible starts out on, its RNA polymerase is working, and so you will have to activate a repressor to turn it off. The inducible ones, they start out off. That's why we can induce them, like you're inducing labor. Person's not in labor, so you need to turn it on. So it starts out as off. 2, we have our RNA polymerase, and it is blocked by an active repressor. That's why it's off. Number 3, we will deactivate the repressor. You don't need to make these genes to break down lactose. So when lactose, so it's always being repressed, it's turned off with this mechanism. So when lactose comes along, it'll bind with that repressor to deactivate it. So it moves off so the RNA polymerase can work. So that's the idea. And remember, operons are only going to be in bacteria. So here, this will, in this slide, describe it in a little bit more detail than what I'm covering, if you would like to read more detail. But there it is, yes. So our little memory thing here is going to be in really important regulations. So if you were to write down in really important regulations, IRIR, inducible, in the inducible operating operon system, it is normally inactive and needs an inducer. Okay, so eyes. Okay, and repressible systems, repressible operons are running and can be turned off by a repressor. So I like that one because if you guys aren't not following what I'm saying out loud, that's okay. If you open up the slides in the notes like area, I have notes in a lot of these slides that might be useful for you guys when you're studying, but that's what I'm referring to right here is what I'm reading from here. So what that actually stands for, I and R. The I's for the inducible and the R's for the repressible. And it has the little sayings that will go with them to help you remember which one's doing what. Just I's for the I and then R's for the R. So our questions for these, which of the following statements accurately describes a key difference between inducible and repressible operons? Let's go through them. In A, we have inducible operons are typically off. I'm sorry. You guys aren't seeing what I'm doing either. And so I'm like, keep saying I'm sorry, like you can see it and you can't. Anyways, inducible operons are typically off and are turned on by an inducer. Okay, inducible operons are turned on by an inducer. This is making sense. This is what we talked about earlier. In the exactly previous slide, I didn't mention inducer in the inducible operon, but how are we going to deactivate the repressor? Well, we said lactose is going to bind it in the lac operon. That was our example. Lactose in this case is an inducer. So I just wanted to go back to that so you understand where it falls in with the steps. So this one is sounding pretty good. All right, B, inducible operons are always on and are turned off by a repressor. No. Inducible operons are not on, they are off. So you're not going to be turning them off. So that's B is wrong. Both of them are regulated by the presence of glucose. What are you? No. And then inducible operons and repressible operons both use the same type of repressor, not a rule. Okay. They don't have the same type. Repressor is a general term describing a group of things that do these actions. Okay. So to be clear, so it has to be A on this one. So inducible operons are off. You're going to induce them by introducing the inducer. Repressible operons are off. And they're going to be, wait, what does it say? I keep seeing off. I keep reading the word off over and over again every time I see it. Repressible operons can only be repressed because they are on. Thank you, Lauren. Repressible operons are on and they are turned off by the repressor. Repressor is going to be doing the turning off always. It doesn't matter which operant we're talking about. If your gene is turned off, it's because of the repressor. Was the repressor there to start with and you're removing it? That's inducing. Or is the repressor getting put on there after the fact? That's a repressible operant. So that's what this is getting at. That's a little better than what I'm trying to say. Sorry, y'all, it is late in the day. It's 4 o'clock almost, and I have not finished eating my lunch. All right, a pharmaceutical company is developing a drug that aims to inhibit the expression of genes involved in bacterial antibiotic resistance. Okay, so the drug specifically targets the repressor protein. So it's a repressor protein that regulates a repressible operon. So if we go back to that and we can actually listen to read the words before we start speaking, it's just me being the one that's having that problem. A repressible is what we're dealing with. So it is something that can be turned off. So it must be on. And we're talking about targeting the thing that can turn it off. So normally repressor turns off that system. Now imagine that it doesn't work. That's the idea of what's going to be going on with this drug. Which of the following, see how I have to draw this out even to like make it clear to me what I'm reading. You guys are allowed to do that on the test, please. So which of the following would be a likely outcome if of this drug's action on the bacterial cell? Well, let's see. Will the operon remain off? No, it wasn't off. It was on. Can't remain off. Already, y'all gotta get on those words. It's lying to you. All right, B, the operon will be activated in the absence of the co-repressor. What? It's not what? It's already on, right? C, the drug will have no effect on the operon since it only targets inducible operons. I mean, to say that specifically, but I think what, hold on. We're going to come back to B here in a second. All right, C, the drug will have no effect on it since it only targets inducible operons. It doesn't say anything about that, so we're going to cross that one out. All right, D, the operon will be turned off regardless of the presence of the inducer, leading to decreased expression. Now, no, it doesn't say anything about turning it off. It actually says that, you know, we wouldn't be able to repress, so stay on. It's going to stay on. So that's why I say come back to B. So let's go back and reread this. Because at first I would like read it and then it like hit me like, okay, wait. Okay, the operon will be activated in the absence of the co-repressor, whatever that means. So this is just talking about the repressor, and we're saying co-repressor, but that's not an important term. So it's talking about the repressor here. The operon is going to be activated, so it's just going to work even when the repressor isn't there. That's what that is saying. That's why it hit me all of a sudden, leading to increased expression. Yes, because we can't turn it off. That's exactly what's going to be happening. I just was tripped up by that co-repressor part of it. My apologies. It just hit me like in the middle of this. So it's that's why. Your questions won't be that complicated because to me, co-repressor is something that I don't expect you guys to know the difference. Like that's the same thing as a repressor. I wouldn't do that to you. I would be clear about it, but just so we know. But the word still might trip you up, so be aware of that. All right, so chapter 8. Now, this is our genetic engineering chapters. What are plasmids? And you wouldn't think I would have to put on this that what a plasmid is, but this is a very commonly missed question on my exams. What are plasmids? Plasmids are extra little bits of circular DNA. So they are circular and they are DNA. They exist separately from the bacteria's chromosome. They're not part of the chromosome. They're an extra thing. They will carry typically genes that are helpful, but not necessary. Usually, if they're keeping them, it's because it's helpful. Okay. And they can switch them between cells via either what they would call the conjugation sex pili. That's say the sex pili, which we learned about. That's little channels between the cells. So that's conjugation. And or you can do can't think of it. Transformation, that's what it is. This is less common in the nature in actual bacteria. They will usually do the sex pili one. Transformation is something that we use to put them into bacteria by our own mechanisms. Even though transformation exists in bacteria, typically with plasmids, it's not something that they do with plasmids, but will force our bacteria to do it with plasmids. Transformation just means you're picking up outside DNA. So a cell next to you dies and its DNA is hanging out in the area that you're in, and you can just pick it up and make it part of your genes. Okay. So here's a little thing to help us remember. It stands for plasmids. Go figure. Personal loops always support microbes in diverse situations. So what this is referring to for personal, that's talking about plasmids are separate from chromosomal DNA. They are their own person, if you will. Okay, personal. They're isolated, if you will. Loops, they are circle in shape, always, because they always replicate independently from the chromosome. They're not part of the chromosome. I don't know how many times I need to say it. Support, they will have benefits that can support the cell, but they are not necessary. They're found in bacteria. M is microbes. And I is can transfer between cells. I in important, I guess is what the I is trying to get at, can transfer between cells. Important for gene transfer if you're trying to share things like antibiotic resistance. D is diverse. So we're talking about a range of advantages like antibiotic resistance. This doesn't want to scroll. And then situations, we can use this for biotech, which has different situations. You can see it in other than in nature. Okay, so that's our little memory thing for that one. Here's our question for this one. Which of the following statements best describes a key characteristic of plasmids in bacteria? A says plasmids are linear DNA molecules that are essential for bacterial division. No, they're not essential. We already know that. They're not essential. And they are not linear either. They're circle. So those are why that is wrong. Plasmids replicate independently of the bacterial chromosome. Yes, because they're a completely separate thing. And they often carry genes that confer advantageous traits, but not required. So this one's looking promising. C, plasmids are large chromosomal DNA segments that integrate into the bacterial genome. No, they exist separately from the genome, and they are going to be circle bits completely separate from that. Little tiny, they're very small in respect to it. Okay, so they're not large. No, they're not chromosomal. No, they're not in the bacterial chromosome. All right, plasmids function as the main source of genetic material for bacteria during replication. What? No, we are just saying they're extra, right? They're not required. Things that are required would be, but this is not required, so it can't be the main source. So our answer is B. All right, A. Oh, sorry, A. The next question. A new bacterial strain has been identified with resistance to multiple antibiotics. Genetic analysis reveals that this resistance is conferred by genes located on plasmids. Okay, so we have plasmids that are conferring antibiotic resistance. What is the most likely mechanism by which these plasmids contribute to the spread of the antibiotic resistance? So how are they spreading antibiotic resistance to the other bacteria? That's what it's asking us. So this is talking about transferring or copying these guys and moving them from cell to another cell. That's what this is talking about. We already talked about conjugation and how you can, basically spread these that way by the sex pili, right? So that's what I'm going to start with looking for, right? Okay, so A, the plasmids are integrated into the bacterial chromosome. Yo, no, not in the chromosome. B, plasmids are transferred between bacteria through conjugation. Looking good, looking good. Okay. We're getting on with B again. Through conjugation, always read it all the way through, right? Allowing the spread of antibiotic resistance genes to other bacterial cells. It's still looking good. The plasmids are degraded by the bacterial cell's own enzymes, preventing the spread of resistance genes. Doesn't make sense because why would you want to prevent the spread of resistance genes? This is talking about spreading them. So that's not the choice because that doesn't even make sense. And then the plasmids are only present in a subset of bacterial cells, so they do not contribute to the overall spread of resistance. Now, we're talking about them, leading to the spread of resistance. So again, once again, same reason as C, but for a different reason here. They're not, even if they were present in a certain subset of bacterial cells, those subsets could spread it to other organisms by conjugation. So that's why B is the answer. That's how they would spread. Next thing is restriction enzymes. Don't you love it? The restriction enzymes, also known as restriction endonucleases, and you might hear it either way. You'd probably be thankful if you heard enzyme instead of endonuclease. But restricted because it is restricted to cutting at specific palindromes. And those palindrome sequences are different for each enzyme of all the enzymes that we know, okay? So those are molecular scissors. Set up molecular scissors, cool. They protect against foreign DNA normally in the bacteria. So characteristics are going to be specificity. We're only going to be cutting at specific palindrome sequences, easy enough. And the cutting action, the one that we were definitely more interested in was creating those sticky ends, so single strand overhangs because of how it cuts. Because remember, if it was A, G, G, C, C, T, and you could fold it over and it would still work that way. And its complementary strand would be T, C, C, G, G, A. And you could make this up if you understand about the rules of it, right? And this is them from one strand to another. So this would be your 5' end, and it would be leading up to this, and this would be going on to your 3' end. And this is your 3, and this is your 5. So you know where the starts of these guys are. Remember that they're anti-parallel. This is your double strand and what they're matching up to. And you can see A, G, G, C, C, T on that top one, A, G, G, C, C, T. And then on the bottom, if you start at the 5' end, A, G, G, C, C, D, palindrome. These guys typically cut like something along the lines of, I'm going to not do circles. I'm going to do it like this. Okay. And then I'm going to scribble over it. So get ready because that's about to happen here. So I hope you wrote down your little note on what the palindrome means. Okay. So you would have one. Come on, dude. I don't even know where my cursor goes. It takes a holiday. There it goes. This would be one of your strands, and this would go on to be double strand over here, right? Double stranded over here. And then the other one, how are we doing it like that? Red again. Okay. And then that other one, obviously, this can be this one. Oh, how is it yellow? I clicked on red. I'm not getting frustrated, y'all. Don't worry. It literally looks red to me. Why are you being a butt? there we go. Okay, so then these would separate out. This would be going on to the rest of your plasmid or chromosome or whatever it is out this way, and the other one to the other side. But you'd be left with these little overhangs of single strand. Those could stick up to things that had that same sequence on that strand, right? Like in this case, it had the GGCZ or something. And so would the other one would be the complement to that. So you'd be trying to match up with things that had those sequences. So if you have two bits of DNA, one from a person, and then one is a plasmid that will go in bacteria later, you can cut them with the same enzyme. They'll have the sticky ends, and you can insert that person DNA in there at that sticky end, and then seal it up with DNA ligase so that it's actually functional. So it's pretty useful concept in genetic engineering, and this slide goes into some detail on how exactly it is useful. We talked about it in class a little bit, but you're welcome to read through this, but I definitely want you guys to be aware of a plasmid, which we talked about in the previous slide, and a restriction endonuclease, which we're talking about in this slide, and what they kind of would be useful for in relation to one another, right? So restriction endonucleases are enzymes made by bacteria naturally to get rid of foreign DNA that they don't want in their cells. They cut at palindromic sequences, like we just went over, and leave little sticky ends that we have hijacked for the use in biotech. Okay. Our little mnemonic is restriction. Real enzymes slice through recorded information, clearly targeting intruding organisms. Oh, they're nucleic acids. Sorry, I left out the nucleic acid. Clearly targeting intruding organisms, nucleic acids. Oh, such a little mnemonic for that one. That one I feel like kind of self-explains itself. But in the notes, if you want more explanation, it is there. All right,
Is
example.
there some place to get a copy of these slides?
Yes, they're on the discussion board on Canvas. So if you go on to our course and then go to discussion, I have each week as we go through, I'll post the slides. So there should
Oh, awesome.
Mhm.
Thank you.
Yeah, no problem. Definitely helps to have them. And there's just too much for me to cover in a review on these, but I want
Yeah,
you
and
to,
it's good info.
yeah. So our questions for this one, which of the following best describes the role of restriction enzymes in molecular biology? So this is talking about how we might use this in science. So A, they're used to amplify DNA sequences. Well, they're not, because they're cutting. Remember, restriction enzymes are going to be, or endonucleases, they're going to be molecular scissors. So they're not amplifying anything. They cut DNA at specific nucleotide sequences, creating fragments with sticky or blunt ends. It's looking good. I know we focus on the sticky, but they can create blunt ends as well. They synthesize new DNA strands by adding nucleotides to a growing chain. They are not making new DNA. They're just cutting it. They bind to specific RNA sequences to inhibit their translation into proteins. Again, these enzymes are not binding to something or dealing with anything like that. We're just cutting the DNA sequence. That's all that's happening there. So it's going to be B. I don't know why I circled the whole thing, but there you go. All right, the next question for this one, a clinical laboratory is using restriction enzymes to diagnose a genetic disorder. The disorder is caused by a specific mutation that alters a restriction enzyme recognition site. So we have introduced that. So when we had before our palindromic sequence, so GG, or let's even put, okay, fine, I wrote that. TCC, G, G, A, and it's going to read the same thing on the other side. Well, if it's going to recognize this sequence and the same thing on the other side, and you're changing it, okay, to that restriction endonuclease, and this disorder is caused by a mutation that alters a recognition site. So normally it could cut here, but now it can't. So how would this mutation affect the results of a restriction fragment length polymorphism? So that's talking about the different tiny little bits of DNA that you get from cutting apart the DNA with these enzymes. And remember the little pictures that we have with those lines that are getting spread out that represents the larger to smaller at the end DNA fragments. that we can get just from digesting the DNA with these enzymes. So if you cut it with this thing that recognizes that sequence, and now we're talking about mutation and it doesn't cut there anymore, right? So now how do we look at it? So would it be A? Sorry, I can't move. The mutation would create a new recognition site for the restriction enzyme, resulting in the appearance of an additional DNA fragment. It isn't creating a new site. And I would expect that if we're removing a site, we're going to have a longer fragment, right? And potentially decreasing the number of fragments because of that. So the next one, mutation would disrupt the restriction enzyme's ability to cut at that site, leading to the absence of the expected DNA fragments. Okay, that's more familiar. So we're not going to see what we thought we would see. That's what that's saying to us. The mutation would have no effect on the enzyme's activity. No, because it depends on that specific sequence. The mutation would enhance the restriction enzyme's cutting efficiency. It would not enhance it or its efficiency. That's not anything we ever talked about with any of that. So it's not that. It's got to be B. Back to that one. Okay, so next thing. Chapter 8. Most missed concepts. What is PCR? Okay, PCR, yes. We went over this one pretty in depth in class. I tried to focus a little bit more on it this semester since this is a commonly missed concept. Remember, our 3 main steps are going to be denaturation, annealing, then extension or elongation is the same words. Okay. So denaturation is separating the DNA strands using heat. So first of all, we should remember that PCR, which stands for polymerase chain reaction, which is something that you do need to know. If I put a word in front of you that says, I don't know, proper can rabies, I don't know what. And then it also is PCR. Be sure that you know what your PCR means compared to other PCRs, okay? So we're just going to amplify DNA. So PCR, amplify, or make copies of. All right, next we got the denaturation step that is separating the DNA strands using high heat. The second step is annealing. So this is binding on our very gene-specific primers, so site-specific primers. You don't want to just amplify any old DNA. You are looking for specific genes to tell you the presence or absence of something, typically, or specific genes to amplify those genes, make copies and copies of those genes so you can use them in your science, right? Site-specific primers are going to bind on. These are starting points as well as making it specific for whatever you're looking at. Extension is just adding on the nucleotides in that area. So building the DNA strands. You guys remember how we've already gone over twice. The original strands are going to separate apart, and then we get the new strand built off of each one as a template, right? Well, it's the exact same thing happening here. This is just targeting a specific part of a gene or part of a chromosome or some sort of sequence specifically. The DNA that you get out of this is not going to tell you anything. It's just going to tell you if you amplified this DNA or not. So if your primers are binding somewhere specific and you're getting amplification, you're seeing increased DNA in your sample, well, the only way it's going to increase the DNA in the sample is if the primers are binding to the right thing, right? So keep that in mind. All right, so there's our summary of the little steps: denaturation, the heat separating, annealing, adding the primers, and extension, adding on the nucleotides. And that is one cycle. All right, so here's to remember: dogs always eat, which is a weird one, but that's our 3 steps, DAE. Dogs always eat. Denaturation, that's your dogs. Annealing, that's your always. And then extension, eat. All right, questions for this one. Which of the following best describes the purpose of the annealing step in PCR? All right, so the annealing step, like we said, we should always, when we're, studying, I'm aware that this is still new to you. We already had the DAE. So this is denature. These are 3 steps. So if I'm asking you about the steps of PCR, you should probably be looking at this: extension or elongation. Whatever helps you remember what is going on there, okay? So we're talking here, what describes this one? Well, we said denature was separating, and anneal is primers, and extension was growing the strand. This is definitely an arrow. All right, so let's look through it. Okay, so annealing, does it separate the double-stranded DNA into single strands? No, it doesn't, because all it is doing is adding primers. B, does it allow primers to bind to their complementary sequences on a single strand of DNA? Absolutely it does. C, does it allow us to synthesize new DNA strands by adding the nucleotides? It's not adding the nucleotides, it's just a primers. Primers are just sitting on their complementary sequence. That's all they're doing. They don't do anything else. And then to amplify the DNA by increasing the temperature, they're not increasing the temperature either, the primers. So annealing doesn't involve those things. Annealing involves the primers. And if you know that, it makes it easy. All right, so the second question, a clinical laboratory is using PCR to detect the presence of a pathogen in a patient's sample, like testing for COVID on your PCR test. If the test is successful, what do you expect in your positive result? So if this person has COVID, what do you expect to see if you ran it in their PCR? Would the PCR reaction produce no DNA products? No, that would be a negative. Would the PCR reaction yield a single large DNA fragment that is larger than you thought it would be? So that's how you know? No, it's amplifying just the sequence that is already there. It's not changing it or making it a different size or anything. PCR reaction generates multiple DNA fragments of the expected size, indicating successful amplification of that specific DNA sequence. That's sounding familiar. It's just a lot more words than I would have used. The PCR reaction results in the formation of a new type of primer. No, it's not going to make anything new. It's just copying what's already in there. So that's how we know it's C. We're just amplifying the sequence if that person is positive. All right, our last thing for the review is gel electrophoresis, which again, we went over that DNA has an overall negative charge because of that phosphate in the backbone, all those phosphate, sugar, phosphate, sugar, phosphate, sugar. It's a lot of phosphates and they are negatively charged. So they have a negative charge in our DNA. And when we put electricity through it, the negatively charged DNA is going to want to move to the positive end. Opposites attract. So the next thing, the bigger the pieces, the big pieces get caught up and the little pieces can move faster. I mean, just imagine it. If you were like climbing through a pile of rocks, can a cockroach get through it faster or can an elephant get through that pile faster? If you're trying to get down to the bottom of it. Obviously the cockroach, but that is the same concept here. It's a size thing. So the larger pieces get stuck up and stay near the top and the smaller ones will be at the bottom. And of course, there's a whole lot of variation in there in the middle. The principles in this one, the gel that we use is agarose gel. This is what we use for DNA and RNA, which is what we're talking about here. And it's a sugar based off of agar that we use like in lab for our media. It's just like a purified version of it. Then we will be able to put our samples into wells that we have put into the gel shape, like literally dropping DNA liquid in there and then running electricity through it. Then the size and the charge are going to cause your DNA to move through and create these little bands that we can visualize using a variety of methods of looking at the DNA, whether it's ultraviolet or whether it's staining the DNA itself. We can look at them on the gel, and that's where we get those pictures from. So JESMs is our thing here. Giant elephants should move slowly. If that doesn't tell you what's going on as far as the size of these guys moving through here, I know what to tell you, but the giant elephants should move slowly. G is supposed to be for the gel. Okay. E is for like the electric field. S is for the size. M is for moving through the gel. S is, slowly, the larger molecules are going to move slowly. So that's kind of what those letters are going to be standing for. All right, our questions relating to this topic: which of the following factors affects the movement of molecules through the gel during electrophoresis? Well, we know electrophoresis depends on, before we even look at the choices, the first thing is going to be the electrical current. And that's going to move our negative DNA to our positive end. And the other thing is going to be the size. And like we said, large, slow, small, fast. Okay, so we already have that down, so we can just refer to it if we need to. So, okay, is it the temperature of the room? What? The color of the gel? No, I don't think we talked about any of that. The size and charge of the molecules, now we're talking, because that's literally what we have over there on the side, or the pH of the buffer solution. Now this one's tricky because this could potentially affect it as well because of how the pH would give a more positive charge to the solution or whatever, but it really shouldn't be affecting the actual size and charge of the molecules themselves. So don't fall for it. It should be C as your answer. That's still going to hold. Okay. A. I did it again. I don't know why I always see this and I think A. Second question. The clinical laboratory, I'm losing it, is using gel electrophoresis to analyze a patient's blood sample. So we're using this whole thing that we already wrote off to the left side. The electric stuff is going to have the negatively charged DNA moving to the positive. And we're going to have the size, the large ones are moving slow and the small ones are moving fast. So we've already still got that concept because of this question. We can say that. So there's a genetic mutation. If a specific DNA fragment appears at a different position in the gel compared to a normal control, so that means looking at DNA for that gene is behaving differently than the gene is for somebody who has the mutation. But it's saying in the reverse of that, but you know what I'm saying. So you have a mutation and your fragments are running differently than the person who doesn't have it, right? And now you can use that as a means to basically determine what's going on with your patient. So what's going on with your patient if they got different movement of their bands? The patient's DNA fragment is the same size? No, the patient's, that's literally what we're talking about over here, right? It can't be the same size if they're at different places. The patient's DNA has been degraded? What? Where did we even mention this? If we're doing this properly, proper science, you shouldn't be having that problem. The patient's DNA fragment has a different size, suggesting a potential mutation, which is why it has a different size. That's sounding like what I literally just said. And then the patient's DNA has a different charge, leading to a different color in the gel. Well, we did not talk about a color in the gel, and that's a ridiculousness. So that already should give it away as this is a ridiculousness. If it had a different charge, it shouldn't. DNA is negative and DNA is negative, and that's the end of it. Mutations aren't going to change that. That phosphate sugar backbone is on everybody's, regardless of your genetic code. Your genetic code is on your ATCG, not on your phosphate sugar backbone. So that shouldn't be it. So that's why C is the answer. Y'all, we made it. Anybody have any questions about any of this stuff for me before we kick off? I'll have this open in case you guys type in it. No questions, no questions. If you happen, of course, to think of any questions, feel free to email me. Or, of course, we have still more time coming up in the next few weeks. You can always bring it up in class. That'd be great too, so everybody can benefit from what you're learning and all that. So if that's it, then I'm gonna finish eating my lunch. I've got another class in about an hour. So, um, so yeah, you guys don't have any questions? I will see you next week. Have a good weekend.